Am I audible or not?
No, ma'am.
Now,
Ma'am, it's very low.
Yes, ma'am.
is it fine now or not?
Yes, ma'am. It's better now.
Better now? Okay, fine. Just let me remove my headset. Am I audible now or not? No.
Yes.
Is it fine now? I've removed my headset. Actually, there was.
Yes, ma'am.
It's fine now. Okay, fine. Okay, so good morning, students. And last lecture we discussed about the pathogenesis. That what is pathogenesis? It is a mechanism which leads to the development of a disease, right? And number of whatever changes or series of changes which takes place in a structure, function, or a cells or a tissues of the host, which are caused due to the pathogen, those changes are included in this, right? So last time we discussed that when pathogen infect host plants, or when it try to establish itself in the host plant, so it secretes number of enzymes, right? Cutinases, cellulases, pectinases, lignases, right? So these enzymes excuse me, they help in the degradation of the cell walls, and to allow pathogen entry into the host. Then apart from this, the pathogens, they also secrete toxins as a chemical weapons. So such as we discussed, that there is a TAP toxin which is secreted by the pathogen, which causes the wildfire disease of a tobacco. And physilotoxin, again this toxin is secreted or involved in the bacterial bean blight, which is called a halo blight also. And 10 toxins also. So there are a number of the toxins which are secreted. We also discussed that these toxins can be the host specific also, and there can be the non-host specific toxins also, which are being secreted, right? So we discussed about basically the pathogen factors, what exactly they are doing, what kind of enzymes they are releasing in order to cause in order to gain entry into the host plants. Now the second part is, right, the pathogen host part, right? So what host is doing to prevent or to reduce the infection, or to avoid the pathogen, if, even if it is entered the host plant. So how it is going to react, how it is going to produce some chemicals, what are the different structural chemicals, what are the certain constitutive or induced responses by the host when a pathogen attack, right? So it is all part of the defense mechanism system in the plants, right? For example, when pathogens, they affect humans, so our immune system, they become active, and then a lot of other things happens, and they try to the first step is try to avoid that we the they try to avoid the pathogen. So in the similarly, the host plants also, they behave in a similar way. They might generate certain chemicals, they might generate certain enzymes. There are some structural defense mechanisms, or structural changes in the plant, which could again affect the pathogen impact on the plant, right? So when we talk about the defense mechanism in the plant, so we say they are constitutive or induced, a structural or a morphological defense, right, or a biochemical defense. So these again, these defenses, they are affected by the age of the plant, type of the organ infected, what is the nutritional status of the plant, and what is the host environmental condition. So all these factors, they affect these kind of this is the timeline of infection. So this we discussed, I guess, in the last lecture, or not last lecture also, but when we were discussing about the infection process, right? So how outside the host and how inside the host, structural and constitutive defenses, how they are outside the host and how they are inside the host, right? So fungal pathogen, they pre-infection is like they are going to germinate, they are going to go for a germ tube formation, aprosodium formation, and penetration pack formation, and then some general induced defenses will take place, maybe some papillae formation, right? And inside the host is a post-infection. What happens? Histological formation, toxin formation, or a detoxification of the phytolexins which are being produced, right? So they cork and lignin layers in the plant, right? So cork and lignin layers in the plants, they also prevent the infection, or they also avoid the infection, or they to some extent they stop the pathogen. Or there could be a systemic acquired resistance. This will study in detail what is systemic acquired resistance, what kind of chemicals are being released when this kind of a resistance takes place. Then hypersensitivity is another act by the host when the pathogen attack them, right? So the hypersensitive response is being generated, which led to the killing or a necrosis of the tissue when the pathogen, or just at the spot where pathogen has gained entry into the host, so that along with the healthy tissues or its own tissues, it's killing, the pathogen is also avoided to move further. So these are the different kind of the responses which plants takes, right? So defense or a resistance. So see, we know that there are certain kind of a genes or a naturally resistant genes which are present in the host plant, due to which the pathogen is not able to infect the host plant. So there are different types of the resistance which are available. Could be the non-host resistance, could be the polygenic resistance, or could be the monogenic resistance, or R gene resistance, which we'll study in detail, right? So what is non-host resistance, right? In this case, what happens when a plant resists the attack of the pathogenic organism, which is otherwise not the host of that pathogen, is termed as a non-host resistant. For example, potato late blight pathogen do not infect apple or a wheat plant, right? Or similarly, apple pathogens such as Venturinia inequalis, it does not attack pathogen potato. So this is a non-host resistance. That means you are not, the particular pathogens, they are going to attack a particular host only, they are not going to attack some other host, right? So this kind of a resistance is called non-host resistant. And similarly, the fungus that causes powdery mildew on the wheat does not infect barley, and vice versa, right? So this is right. Then this is a non-host resistant. Potato late blight pathogen cannot infect apple, or Venturinia inequalis cannot infect the potato, right? Then the host defenses, as we said, constitutive, or they can be constitutive or induced. So constitutive में again structural and biochemical responses आते हैं, and induced may be again structural and biochemical. So in constitutive responses, structural में there will be a wax, there will be cuticle thickness, there will be hairs, there will be a shape and activity of the stomata. Biochemical responses could be some preformed inhibitors or some phenolic compounds which are being generated by the plant. Then when we talk about the induced responses, again the corky layer or abscission layer which is being formed. The biochemical include this hypersensitivity or a phytolexins, or a systemic acquired resistance, or antimicrobial enzymes, right? So these kind of responses are the biochemical induced responses by the host which are being generated, right? So plant defense responses, when we talk about constitutive responses, means physical barriers or a or could be, that means the structural changes in the plant, or could be biochemical, certain kind of the chemicals which are being released. Inducible, again structural में आपका corky layer आ जाएगा, आपका abscission layers are being formed, आपका tylosis form होते हैं. Biochemical में chemicals such as phytolexins हो गया, आपका generate secrete होते हैं. Then आपका hypersensitive response हो गया, आपका systemic acquired resistance है. ये सारे के सारे आपके biochemical responses आते हैं, right? So structural defense could be your pre-existing or pre-infectional structural barriers, could be the surface barriers such as presence of waxy layers. Due to those layers also, the pathogens, they are unable to enter. Hairs or the lignin, thickness of the cuticle, for example, in resistant flax species, the thickness of the cuticle is more. It is for example, it is not affected by this Puccinia graminostatici, right? So the thickness and toughness of the outer wall of the epidermal cells are apparently important factor in the resistance of some plants to certain pathogen. Why? Because they are so thick, their cuticle is so thick, or their outer wall is so thick that the pathogen is not able to gain entry. वो enter ही नहीं कर पाता, या pre penetration process होते हैं वो ही नहीं हो पाता है. So that means it is not affecting the plant. So plant is resistant to that particular pathogen. So these factors are playing role here, right? And other important factors could be the size of the stomata also. For example, in resistant citrus varieties, they have very small stomata, so pathogen cannot enter. Then location and shape of the stomata, opening and closing of the stomatas, these things also play a very important role. Then post-infectional defense structures. What are the post-infectional defense structures? वो आपके pre-infectional defense structures थे. These are the post-infectional. So histological defense. For example, there is a tylose formation. Example in case of a wilt. Corky layers. Example in case of a potato, which is being affected by Rhizoctonia solani. Then there are cellular defenses, which means altered walls of the cells or a changes in cell wall thickening. Deposition of callous papillae in the cell wall so that pathogen उसके अंदर ना और अंदर ना आ पाए. Then cytoplasmic defense include your hypersensitive response, which is being shown by the host. So what is this hypersensitive response? So it result in the sudden death of the host cell in the vicinity of the pathogen. Means जहाँ पे pathogen ने enter किया host plant के उसके around, it will, the host response will result in the killing of the healthy cells of its own cells of the plants also, right? Highest degree of resistance. Again, then both structural and biochemical in nature. So it is very common in obligate pathogens like fungi, viruses, and nematodes. And it is found in other fungal and bacterial infections also. And due to this hypersensitive response, the necrotic tissues, they isolate the obligate pathogen from the living cells, then devoid the pathogen of the nutrition and thus starve and die. Because at the end pathogen will also need healthy cell to multiply and grow. अगर उसने उसके आसपास के सारे cells around its vicinity of that pathogens, these are being killed due to the hypersensitive response, the pathogen will also starve, right? And it will also die because it is not getting any healthy tissue to further multiply and survive, right? So this is again the example of induced structural defenses are abscission layer in Xanthomonas pruni, which causes short hole, then cork layer in Rhizoctonia solani, which causes canker in potato. So the formation of a cork or abscission layers can limit the size of the lesions and consequently the extent of damage that can be caused by the single infection. Again, they provide protection by inhibiting the further spread of the pathogen. So it prevent the further spread of the pathogen, it block the spread of the toxic substances of the pathogen, and it stop the flow of nutrients to the infection point so that pathogen, it does not get any nutritions or nutrients also to further survive. Then there are induced structural defenses which include tylosis. So what are these tyloses? These are actually the overgrowth of a living cells that protrude via pits into the xylem vessels and it block the vascular system. Now, if they form abundantly and very quickly, they can spot. Now, it's definitely जो vascular pathogens होते हैं, pathogens which spread through the vascular system, so those pathogens can be stopped there, right? So if they form very quickly and abundantly, they can stop the spread of the vascular wilt pathogens, mostly wilting pathogens or a wilting which is caused due to the fungal or a bacterial pathogens. Those pathogens can be stopped here. Again, this formation is triggered by the stress-like conditions. Then there are biochemical defenses, right? So pre-existing biochemical defenses are basically inhibitors which are released by the plants in its environment. It could be the fungi toxic exudates of tomato and a sugar beet. For example, they inhibit germination of botrytis and cercospora. Then oil capric acid on elm seed toxic to ceratocystis almi spores. Then phenolics, which are again secreted by certain kind of a plant, especially the जो आपके जो ये temperate trees या fruit trees होते हैं, they have lot of phenolics, so they secrete lot of phenolic compounds. Then again in onions, red scale onion, again smudge Calectotrichum, pathogen Calectotrichum. So this can be used. Phenolics can also be helpful, or they also act as they are being released as a biochemical defense mechanism. Then there are inhibitors which are present in the cells, which are being stored in the vacuoles or in lysogenous glands, in the heartwood of a peritoneum of a plants. Phenolics in the onion, for example, right? Saponins, again tomatin in tomato. So there are a number of compounds which acts as the inhibitors which are present in the plant cells. Some plants also contain hydrolytic enzymes such as gluconesis, chitonesis. So they also somewhere help in the defense mechanism. Then there are these are the example, again the same, the constitutive defense, biochemical defenses. So they say that there are two type of the onions, the white type of or yellow onions, which do not contain significant quantities of certain phenolic chemicals, so it is susceptible to the pathogen. Whereas this red type of the onions, they have phenolic compounds, and which make them resistant to the onion smudge disease, or a disease which is caused by some pathogen, right? So in pre-existing biochemical defenses, these defense through lack of essential factor could be due to the lack of some essential factors such as lack of recognitions, or due to the lack of host receptor and sensitive sites for toxin in the host, right? So lack of essential nutrients for the pathogen. Example, some pathogens, they prefer high sugar content, like Helminthosporium oryzae, right? So these pathogens, if these components they are not present in the plants, or there's less of these kind of a components, so these pathogens they are not going to infect those kind of a plants. So defense through lack of essential factors. Then we have a post-infectional biochemical defenses, which include your hypersensitive response. This is the main one, right? Then biochemical inhibitors, which are produced in response to injury by the pathogens, such as phenolics, or oxidation products of the plant, such as phytolexins. So these are the post-infectional biochemical defenses. So induced biochemical defenses by hypersensitive response is the localized death of the host cell at the site of infection, and it is the result of specific recognition of a pathogen attack by the host. So if a host recognize that there's a pathogen attack, right, it is able to recognize, so it generate this hypersensitive recognition. So this is somewhere related to the programmed cell death जो आपके humans में होती है, या फिर हम these are being noticed in the humans or the animals, that there is a programmed cell death. It is just similar to that. The hypersensitive response is again the culmination of the plant defense responses, which is initiated by recognition of the plant specific pathogen and produced signal molecules known as elicitors. So they generate certain kind of a signal molecules which are called elicitors. Now recognition of the elicitors by the host plant, it activates the cascade of biochemical reactions. So when the host plants they recognize these kind of elicitors, certain kind of a biochemical reactions will take place, right, in the attacked and surrounding plant cells, which leads to new or altered cell functions, or and to new or greatly activated defense related compounds. Now the most common new cell function of compounds include a rapid burst of reactive oxygen species. Again, this will lead to increased oxidative reactions, increased ion movements, especially of potassium and hydrogen ion through the cell membrane, and disruption of membrane and loss of cellular compartmentalization, right? So this will lead to increase in certain oxidative reactions or a rapid burst of a reactive oxygen species, and production of antimicrobial substances such as phenolics or phytolexins, and formation of antimicrobial so-called pathogenesis related proteins. So this we call as a PR proteins, pathogenesis related proteins, which are being secreted in response to the pathogen attack in the host plants. Then defense through increased level of phenolics. This also we discussed in the last, I guess. So plants which contains aromatic acids which act as precursors of phenolic compounds. Simplest compound is a phenol, which has a single OH group or on the benzene ring, right? So when benzene ring has more than one OH group, we call it as a polyphenols. So lignins हो गया आपका, phenyl glucoside हो गया, या flavonoids हो गया, या anthocyanin हो गया. These all are polyphenols basically. So this is how. So in plants, the phenolic synthesis takes place through ischemic acid pathway and acidic acid pathways. Don't remember the pathways, because pathways we are not going to discuss in details, right? So we are mostly focusing on what kind of a chemicals are being generated, or what kind of a host responses will lead to generation of certain chemicals which will impact the pathogen. So we are not discussing the mechanism here, right? So phenolic compounds which are produced by plants are caffeic aid, sweet potato, chlorogenic acid in again sweet potato, potato, carrot, fluoretin in apple, and hydroquinone in sweet potato. So these are the phenolic compounds which are being generated by the plants, host plants, in response to the pathogen. Then we have a phytolexins. Again, then when people were studying about the potato late blight interactions, so they defined these phytolexins. They were defined as antibiotics which are being produced in plant pathogen interactions, or maybe they are produced as response to injury or physiological stimuli also. So these were defined phytolexins as a low molecular weight antimicrobial compounds, and these compounds are usually produced or synthesized by and accumulate in the plant cells after microbial infection. So these plants are basically generated or being released by the plant after the pathogen is infecting a particular plant. Again, these plant phytolexins, they play a role in the elicitors in their productions, and not produced during the biotropic infections. Many phytolexins have been isolated from plants, around more than 20 families. For example, Leguminosae or Solanaceae, Malvaceae. So in around more than 20 families, these phytolexin compounds have been isolated. What are the characteristics of phytolexins? So they say that they should be fungistatic, that means they should kill fungus and bacteria also, and they are very active at a very low concentrations also, and these are produced by host in response to infection, or some as a metabolic byproducts of a microorganism and their stimuli also, right? These phytolexins, they are absent in healthy cells, or if they are present in a very small amount, so their concentration increases to many fold when the plant is infected by certain pathogens, right? Usually they remain close to the site of their production. They are produced in quantities which are proportionate to the size of the inoculum. So if inoculum size is more, so they are being released more. Then produced in large quantities in response to weak pathogen or non-pathogen than the virulent ones. Produced relatively very quickly in cell after the infection, just after the infection they are being produced. They are host rather than pathogen specific. So these compounds are not pathogen specific. These are being what does that means? That means they are being released even if any fungi or even if any bacteria is going to infect. So they are host specific rather than the pathogen specific, and different hosts might produce different kind of a phytolexins, right? Best studied phytolexins are legumes. We have a phaseolin. Common beans we have a vestitol. Red clover we have a phaseolodin. Common bean we have a medicarpin, right? So we have a number of phytolexins which are being isolated from the different host plants, right? So phytolexins, this is just an example that they are being synthesized in response to fungal pathogen, in response to the pathogen attack. basically. So some pathogens counterattack producing enzyme that degrade the phytolexin. So this is also, so this is just an example. They've shown that how fungus, if any fungus is being infected, so how the phytolexins are there being released by the cells. Then induced biochemical defenses may we have a systemic acquired resistance. So systemic acquired resistance is the activation of the defenses in distal or a non-infected parts of the plants also. Maybe एक जगह pathogen enter किया है तो systemic acquired resistance is may be activated in some other part of the plant also. Right, so the plant become more resistant to the secondary challenge or inoculation. That means first infection में तो pathogen enter कर गया, but second time it will enter, then the systemic acquired resistance will become activated. So what is systemic acquired resistance? Again, this is a broad-based resistant to different pathogens, right? For example, if there is a primary inoculation with a fungal leaf spot pathogen, it reduces the susceptibility of the host plant to the other fungi as well as to the bacterial and viral pathogens, right? So how does this happen? The salicylic, there's a compound or there's a component called salicylic acid. So this salicylic acid is a part of signaling pathways which are involved in transmission of the defense responses throughout the plant to produce the systemic acquired resistance. So they generate some signals, basically. Salicylic acid, when it is being released, it generates certain signals which sends signals throughout the plant to produce systemic acquired resistance. Now this has led to development of a synthetic chemicals that mimic the role of a salicylic acid. So certain synthetic chemicals are being also made this way, right? So in the induction of systemic acquired resistance, how does it happen? What happens after that? So there's a production of hydrogen peroxide or number of antioxidants. So hydrogen peroxide, we know it is being associated with secondary induction of systemic acquired resistant, and it direct toxic activity to the invading pathogens. So it will the number 2 will be the thickening of the plant cell wall. So this will lead to the production of phenolics and certain ligenic and tannic acids, which will strengthen the walls and inhibit the pathogen enzymes. Then third would be the accumulation of pathogenesis related proteins, or we call them as a PR proteins also, such as chitinases or beta-1,3 glucanases, etc. So these enzymes, they accumulate in the vacuole of the plant cell, and upon attack they directly degrade the fungal cell walls. Again, their action will result in the release of fungal wall components that will elicit the additional defense reaction in the host plants, right? Then the signal compounds which are responsible for induction of PR proteins are salicylic acid, ethylene, xylanase, the polypeptide system in or jasmonic acid, and other polyacrylic acids. So these they basically they are responsible for the induction of the PR proteins. Then pathogenesis related protein, again the PR protein we are talking about. So they are a group of plant coded proteins. They are structurally very diverse and they are toxic to the invading pathogens. And these type of a protein are being generated in the plant when they are under stress by attack of certain kind of a pathogens. Again, the pathogenesis related proteins, they are also known as the B proteins. Some workers has named them as the B proteins. Again, these proteins, they are widely distributed in very trace amount, means smaller amounts, but then these are produced in very large number or high concentration when there is a pathogen attack or when there is a certain kind of a stress in the plant. Now the PR proteins, they again exist in plant cells intracellularly. They are acidic in apoplast and basic in a basic form in vacuoles, and they are also found in intercellular spaces, right? So varying types of a PR proteins have been isolated from the several or a number of different crop plants. So groups of a PR protein, there are the several groups of PR proteins, they are being classified according to their functions or their serological relationship or their amino acid sequences or their molecular weight, or basically according to their different properties. So these are either extremely acidic or they are sometimes extremely basic. So they are very highly soluble and very reactive. The better known PR proteins are PR1 proteins, which are B13 glucanase, chitinase, lysozymes. Then PR4 proteins, They are osmitin-like proteins, they are cysteine-rich protein, they are glycine-rich proteins, or they are proteinase inhibitors, right? So there are numerous isoforms of each PR proteins in the various host plants which are being found. Although again here also the healthy plants, they may have some trace amount of a PR proteins, but attack by the pathogens or a treatment with elicitors, wounding or a stress induces transcription of a battery of a genes that codes for a PR protein. So their concentration is increased when they are in a stress like condition, when there is a pathogen attack, right? So this occurs as a part of massive switch in the overall pattern of the gene expression, during which normal protein production nearly ceases. Now, the signal compounds which are responsible for induction of PR proteins are salicylic acid, ethylene, xylanase, and polypeptide cysteamine and jasmonic acid. Then significance of a PR protein, again we've seen that they have an antifungal and antimicrobial activity. So they have a very strong antifungal and antimicrobial activity, and they also inhibit spore release and the spore germination also, right? So these have a strong pathogen activities against the pathogen. Then some of the PR proteins such as B1, 3-gluconase and chitinase, they diffuse towards and affect the chitin supported structure of the cell walls of several plant pathogenic fungi, whereas lysozymes, they degrade the glucosamine and murmuric acid components of bacterial cell walls. So they basically they just affecting the bacterial cell walls. They definitely their cell walls are affected or broken. So in a slow course or after a certain times, the bacteria, the pathogen is going to die. Then plants which are genetically engineered to express chitinase genes, they show good resistance against the soil-borne pathogens such as Rhizoctonia solani, or the signal molecules that induce PR protein synthesis. They again seem to be transmuted systematically to other parts of the plant to reduce the disease infection and intensity in those parts for several days or even weeks. So basically, when these kind of a proteins, see, they are being not produced throughout the plant. They are being secreted or produced at one part of the plant, and then they are being taken to the other part of the plant where the pathogen attack is being more, or where the pathogen has attacked, right? So it is not that they are being generated only at the point where the pathogen is attacked. So it is a part of a systemic acquired resistance where pathogen may attack at one point of the plant, but the systemic acquired resistance become activated maybe in the other part of the plant also, to stop the pathogen from the movement further, or to do whatever, so that the infection or pathogen is not able to multiply in the host plants, right? So this was basically about the this whole module was basically about the host responses to the pathogens, and pathogen responses when it affects the host, right? So there's a when pathogen is affecting a host plant, the pathogen will try to multiply as much as it can. It will try to release as much as chemicals or number of toxins so that it can get entry into the host plant, whereas the work of the pathogen or the work of the host is that it can do whatever to stop that pathogen to enter that particular plant, right? So releasing of certain structural defenses, or releasing of certain biochemical defenses, releasing of phytolexins or a PR proteins, or a systemic acquired resistance, or hypersensitive response. So they are going to do, host is also going to do whatever it takes to stop the pathogen. So they definitely it is kind of a survival of the fittest. It comes in that theory only, that if plant is successful in preventing the pathogen to move further, right, by maybe hypersensitive response or SAR response or other chemicals, so that means that plant is resistant to the pathogen. At the end, we'll call that, right? But if pathogen is successful in invading the host and causing symptoms and the losses at the end, so that means the plant is, despite of all those facts, the plant is susceptible to that particular pathogen, right? So this fight, it keeps on going, and plants, they'll try to save themselves from the pathogens. They'll try to invade as much as host as possible to cause the diseases and to perpetuate further, right? So this is how the evolution, it keeps on going, right? Okay, so this was all for today's lecture. If you think that there is something you need to understand more, or there is something you need to understand more about the resistant mechanism in the plant, then we can have one more lecture. See, resistance, when we talk about the resistant in the plant, so it is again itself maybe 2-3 topics, because we need to cover, there's a horizontal resistance, there's a vertical resistance, or there's a monogenic resistance, or there's a polygenic resistance, and there are a number of theories which are being given, that how the plants, their resistance, how the resistant factors are being introduced into the plants, they are being found in the plant, and why they are naturally being found in the some plants, why they are not found in the all the plants. So this is again a very elaborative topic, which is not part of this course curriculum basically. So we just I just try to touch or explain what is the host resistance, non-host resistance, right? What is how different pathogens, they are not able to affect all kind of the host. This is because of the non-host resistance, that they are not going to affect, for example, apple pathogens, they are not going to affect the potato plant, or a potato pathogens, they are not going to affect the apple trees, right? So there are different kinds of a mechanism for this also, and it is a very elaborative study, that how this resistant act, how this resistance is being introduced. There are several theories which are being given for the resistance mechanisms also, right? So that we are not going to discuss. So we just want to focus on basically two important things here, that previously, that what are the structural defenses, structural and biochemical defenses which are being used by the host, and what kind of enzymes are being secreted by, or enzymes, toxins, or the growth hormones, growth factors, they are secreted by the pathogen to infect a particular host plant. So these two were the main components of this particular module. So I just tried to discuss this in the simplest possible way so that you understand, but still just go through these topics once again, just try to read them carefully, and if you find any other related topic on the internet, or maybe a paper, so you can just go through those papers also to understand these things better, right? Fine, so we'll stop here today, we'll not go further, and maybe just one small question I'll publish in your assignment section as a part of your class assessment. Okay, there's just two small questions, just quickly do them and come back.
Ma'am, I actually had a doubt regarding yesterday's test.
Yeah, please go on.
Ma'am, who is the second Roman god over us? Because I could remember only one.
I guess there were two, Robigus and Robigia. I guess just a moment. Robigus and Robigia. They were two god basically of similar name. That's why I hinted them that there was a Robigo and Robigus.
Ma'am, but then wasn't the god Robigus and then there was some festival
Yeah,
named
there was a Robigilia, which was a festival,
after?
and this was for the same basically this rust diseases, right? But God, they were two gods, Robigio and Robigus.
Okay, okay, ma'am. Yeah, thank you.
Students, I have uploaded uh, 4, I guess, PDFs, right? So those 4 PDFs are actually the elaborative form of plant pathology that we are going to study. We have studied in this course particularly, but they are very elaborative, right? Just go through them, and I guess you can just tell if you want to study some extra thing about that what we are studying right now. So those things also you can find there. So I've just uploaded PDFs for the reference purposes. You can refer to those PDFs, right? They are बहुत डिटेल में है, but still you can go through them, right? Okay, so for attendance, please switch on your cameras. Adhish Mishra, Ayushi Jain,
Present, ma'am.
Sam,
Present, ma'am.
Devanshi,
Present, ma'am.
Ishita Arora, yeah, she's there. Janvi Singh,
Present, ma'am.
Kshitij, okay, and Manaswani, yeah, Manaswani is there. Namrata,
Present, ma'am.
Nandini,
Present, ma'am.
Nehal,
Present, ma'am.
Rupal, Sahiba,
Present, ma'am.
Sanjana,
Present, ma'am.
Shakti,
Present, ma'am.
Shivani,
Present, ma'am.
then we have Tanvi,
Present, ma'am.
Tridiv,
Present, ma'am.
Vivek,
Present, ma'am.
Yukta,
Present, ma'am.
Yuvraj.
Present, ma'am.
Okay, students. So I'll see you. We have class now next week, right? Monday. I'll see you on Monday. Thank you. So just go through these things, right? Go through the PDFs that I upload just to understand more. You'll get better understanding, right? Okay. Thank you. I'll see you on Monday.
Thank you, ma'am.
Bye bye. Thank you.
Thank you, ma'am.